everybody, welcome back to our lecture on the central nervous system and specifically the brain. And so now the previous video went over most of the cerebral cortex. Now we're going to travel through the center of the cerebral cortex and then head into the diencephalon portion of the brain, the midbrain, and then also the brain stem. Okay, so you can see from the cross section of the picture here, we're still in the cortex of the brain. But as I had mentioned before, that there's a portion of the cortex of the brain, or I'm sorry, of the cerebrum, in the middle of the cerebrum where the white matter is, there are little pockets of gray matter. So all those little spots that are there, and they're labeled on this slide. You don't have to memorize these labels. But they are referred to collectively as the basal nuclei. And depending on what source you're reading, they might also be referred to as basal ganglia. But the more proper name is basal nuclei, because this is just a cluster of the cell bodies within the central nervous system. Okay? And remember, the cell bodies form the gray matter. Ganglia are a cluster of cell bodies in the peripheral nervous system. So that's why it's not quite the proper name, but it's a name that was labeled years ago, and so some people still use it. So just make sure you understand. Okay, so you can see the basal nuclei, mainly the caudate nucleus, the putamen, the globus pallidus, right? So basically, these are the portions that inhibit unwanted movements. Okay, so when you are moving your muscles, there's actually some stimulation of muscles that you don't want to move. And so this controls for that, and it's got an inhibitory role in motor control. So it kind of selects and maintains purposeful motor activity and suppresses the useless or unwanted patterns of movement. Okay, and so it really is helping to monitor and coordinate slow, sustained contractions, that especially things like related to posture and support. Okay, I think we'll leave it at that. The other thing on this slide I want to point out, and you can see it's pointed there, the corpus callosum. So the corpus callosum doesn't have to do with basal nuclei, but I just wanted to make sure I pointed this out, is it's the white matter tracts that connect the left and the right side of the brain. So earlier I had mentioned that the left and the right side of the brain coordinate with one another, and that's the main way that they do that. Those are axons that are bringing information from each side of the brain to the other side of the brain. Back to our basal nuclei, just to bring up, a type of disease. This is the region where Parkinson's disease is located. Okay, and so it's just over time a gradual destruction of neurons, and these are the neurons that release dopamine. It comes from a particular basal nuclei, and without enough dopamine, then you get symptoms like increased muscle tone, which is this rigidity, right? Involuntary or unwanted movements, and so that's why we call it resting tremors. So someone might be resting and their hand is shaking somewhat. Okay, they might have slowness in initiating and carrying out motor behaviors, right? So it's hard. You might want to stand up, and it takes a while to finally get that coordinated and stand up. And then they end up standing up very slowly. Sometimes if you're doing an ongoing activity, it's hard to stop that. So this is a disease of the basal nuclei, and you can see in the picture here, the part that's lit up red, that is showing the areas that's being affected. Actually, that is showing the areas that are able to function in a healthy way. You see that before, so the person in the middle has Parkinson's, but they're not showing symptoms yet, so they have some functioning. But all the way over on our right side, they have Parkinson's disease, and that red is gone. It means those cells aren't functioning. And those particular cells are called the substantia nigra, and they're not producing dopamine. And so, again, it's dopamine that's helping to control those movements. All right. So now we're going to go to the next portion of the brain, and I wanted to put this picture in here to show that we're out of the cerebrum, but we're in a portion of the brain that sits right in the middle of the cerebrum. The cerebrum surrounds it. So this is called the diencephalon, and here's from a cross section of the brain to see where that diencephalon is. You see the corpus callosum kind of forming an arc along the top, and then the diencephalon is in that middle portion, and it consists of the thalamus, the hypothalamus, and the pineal gland. Okay, and then the pituitary gland is connected to the hypothalamus. We're going to talk a lot about the hypothalamus and the pituitary gland when we do the endocrine system. Okay, but in general, just to know right now, the hypothalamus controls things. So we said that was the main integrating center for homeostasis. So it controls these homeostatically controlled variables like body temperature, thirst, urine output, food intake, right? You can go on and on. Again, we'll talk more about how its effect in the endocrine system and with the pituitary gland. And it also has an effect in the autonomic nervous system, which we will talk about later. And then the pineal gland really plays a role in, I'm sorry, I put the pineal gland back, it participates in our sleep and wake cycle. So that's very important. Okay, one portion I do want to talk about is the thalamus. I mentioned it a little bit earlier because it's all sensory input except for possibly smell. It synapses in the thalamus on its way to the cortex. And so we call it kind of a relay center. It can relay the sensory information coming in and send it to the appropriate area on the cortex. And on its way, it can screen out insignificant signals. So again, I think I mentioned this in an earlier class. When you're focused on something, you don't hear the other ambient noises going on around you. And that's the thalamus helping you to do that. It's kind of blocking out those other signals that are coming in. Okay, so that's really important. And then it routes that important sensory information to the important areas of the cortex, right? And it can help direct your attention then to that stimuli of interest. And there are other parts of the brain that work with that as well. Even the brainstem and the cortical association areas do that. Okay. It also gives you a crude awareness of the various types of sensation, and that's what I talked about in the earlier video. So again, that thing in your hand, this thalamus will let you know is something cold and wet and shaped like a square, right? But then when it gets to your parietal lobe, that's where you put a label on it. Well, this is an ice cube, okay? And it also helps give some degree of consciousness. It does things like positively reinforces voluntary motor behavior initiated by the cortex. So even some information comes from the motor areas out the thalamus. Okay, so a lot of things that the thalamus does there. Another portion of the brain, it's actually several portions of the brain, but together we refer to it as the limbic system. The limbic system is responsible for things like emotion, certain behaviors like basic inborn behavioral patterns. That's more related to survival and perpetuation of the species. So things you don't really have to learn, right? You know, if you see water, you should drink water. Those are not things that you have to learn. And so you can see all the different parts of the limbic system there, the cingulate gyrus, the fornix, portion of the thalamus. The amygdala is associated with fear memory. So when you have a fear, it's within the amygdala, so on and so forth. The hippocampus, though, is really important in memory. Okay, and so I want to go over a little bit about memory next. Okay, so memory, here's all different types of memory. Memory is defined as a storage of acquired knowledge for later recall. Okay, and a memory trace then is the neuronal change responsible for the retention or storage of knowledge. So neuronal change. So when you're forming a memory, you're actually changing your neurons, okay, and making more connections and sometimes new connections. So that's the memory trace, sorry. We have short-term memory. That's just memory that lasts for maybe seconds or hours. So just things like you're going to stop at the grocery store and you have a list in your mind, right? 2 weeks from now, you might not remember the specifics of that list. You know, maybe you remember milk because you always get milk, but you won't remember the specifics because it's unimportant. You don't need to waste brain power on that. So that's short-term memory. Long-term memory is what's retained for days and months and even years, right? And that's hoping what you're doing now. So the information you're absorbing now will first go to your short-term memory. And then what we need to do is we need to consolidate that memory. It means processing it and transferring it to the long-term memory areas where it's stored. Okay? So we're going to actually look at that in a minute. And then there's also something called working memory. So it's just something, again, temporarily holds and interrelates various pieces of information related to a task you're doing right now. Okay, so that's called working memory. So in order to integrate a memory from short-term memory to long-term memory, and this is something your brain is hopefully going to be doing very soon after you watch this, that's a process referred to as long-term potentiation. Now look at this slide. This deals with a synapse. So I think I'm probably going to want you to know the steps of long-term potentiation. Okay, if you go to the PowerPoint slides on Canvas, they actually list out the steps for you there. But so again, so what we're doing here is information that's coming in, then a neuron is stimulating another neuron, and that's where that storage of memory is coming in. Okay, we think this is a type of synaptic learning. And the more that you produce the synapse, what'll happen is the more receptors on the postsynaptic cell will develop and the more dendrites will develop, and then that will make the recall of the memory easier. So we believe by you thinking about and studying, what you're doing is causing this synaptic pathway to occur again and again, which then causes, again, that neuronal change, the growth in those cells, and that is a memory storage. Okay, so first thing you need to know is in the presynaptic cell, and this is mainly what's occurring in the hippocampus. So the presynaptic cell there, the neurotransmitter involved is glutamate. You need to know that. And glutamate binds to receptors on the postsynaptic cell, and there are two types. One is called an AMPA receptor, and the other one is called the NMDA receptor. So you can see that they do two different things. Look at the purple one, the AMPA receptor. This receptor is also a sodium ion channel. So when glutamate binds, sodium enters, we get depolarization. And you can see that in the first picture on the left. Okay, then so sodium diffuses in, you get an EPSP. That's our depolarization, right? That EPSP then helps to prepare the NMDA receptor. So again, on the picture on the left, when you look at that receptor, the blue one, there's an ion of magnesium stuck inside of there. That blocks it. That's what closes the receptor. Okay, calcium can't get in. So two things need to happen. One, the depolarization needs to occur to help kick that magnesium ion out, because depolarization makes the inside more positive, and look, that ion is positive. So it kicks it out. But then also the binding of glutamate allows it to open further and allows calcium to enter. Okay, so calcium flows through based on its concentration gradient, and it stimulates a second messenger pathway. Okay, it's that second messenger is going to stimulate two protein kinases. Okay, and that's going to cause two different pathways to occur. Okay, so one of the things that one of the protein kinase does is it ends up stimulating the DNA to create more AMPA receptors. So more AMPA receptors get inserted in the postsynaptic cell membrane, so therefore it becomes more sensitive to glutamate. And it actually depolarizes more quickly, right? More receptors means more sodium ions that can enter. And so it can depolarize more quickly. That's more sensitive. The other pathway, going to protein kinase 2, what that does is it releases a paracrine signaling molecule. That paracrine molecule is actually nitric oxide. And nitric oxide gets released, and that stimulates the increase of glutamate to be released from the presynaptic cell. So remember, a paracrine just stimulates the cell adjacent to it through the interstitial fluid. That's what nitric oxide is doing. Okay, it's not a neurotransmitter. The neurotransmitter is glutamate coming from the presynaptic cell to the postsynaptic cell. This is why I made you study all of that back in the previous unit. Almost everything you learned in the previous unit is coming into play in this pathway. Okay? The other thing that happens is that the more calcium then that enters the postsynaptic cell, it causes the growth of more dendrites on the postsynaptic cell. And each one of those dendrites now can make a connection with the presynaptic cell and have this synaptic response occur, right? Making it much more sensitive. That's you recalling the memory much more easily. So when you study and you put the work down for a long time and you don't go back and look at it, you're not stimulating this pathway. Okay, and that's why later on, days later, it's really hard for you to recall. I've actually had students tell me, you didn't go over that in class. Yes, I did, and I can prove it to them. And then also, oh my gosh, I don't remember that at all. That tells me you're not looking at the material enough. And not just reading it, but you've got to think about it. Do I really understand why nitric oxide is a paracrine? Well, what was our definition of a paracrine? Again, it was just the molecule that stimulates an adjacent molecule through the interstitial fluid, and that's what's happening here, right? Why is sodium entering, right? Make sure that you compare that back. Well, what is a chemically gated sodium ion channel, right? Well, it's stimulated here in the case of glutamate, which is a monoamine or an amino acid neurotransmitter. Okay, so all of these things you learned already. So make sure you go over it. If you need help, come see me. Long-term potentiation. Okay, from there, we're moving out of the covering of the cerebrum. We're moving out of the diencephalon, and now we're at the back portion of the brain to that portion called the cerebellum. So the cerebellum literally means little cerebrum. It's structurally similar, although being smaller. It's also bilaterally symmetrical, so it has a left and right hemisphere. Okay, it's got an outer cortex. It's got white matter on the inside. Remember the arbor vitae from anatomy. Okay, and it's got nuclei inside of it, just like we saw the basal nuclei in the cerebrum. Okay, but here the cerebellum functions in the coordination of your muscular movements and also balance. And it's also coordination of your eye movements and body movements as well. Okay, we're going to get more specific because what would physiology be without the specifics? Easier, I'm sorry. Right? So let me show this to you because there are basically three functional areas of the cerebellum. Okay, and so that top portion is called the spinocerebellum, the purplish color, pinkish purple, right? You should know the functions of each of these three different areas, right? So what this does is enhances muscle tone, right? Coordinates skilled voluntary movements, and basically, to translate that, it ensures the accurate timing of various muscle contractions to coordinate movements involving multiple joints, right? So movements of your shoulder, your elbow, wrist joints, they must be synchronized just to even pick up a pencil and write. Okay, so all of those movements must be synchronized, and that's the spinocerebellum doing that. Now, yes, that's a voluntary movement, and you're thinking, what about motor cortex? Yes, so that information is also being initiated by the motor cortex, but it's the cerebellum that is fine-tuning it for you. Okay. Let's see. You can think of it as middle management, right? So it's kind of comparing the intentions of what you want to do coming from the motor cortex, and then just coordinates all of that for you, right? So for example, like when you walk, that's a voluntary movement, but you don't consciously think about each step. So that's the cerebellum helping you with that. The bottom portion, the largest portion, the blue area there, that's the cerebrocerebellum. So it says it plays a role in planning and initiating voluntary activity by providing input to those cortical motor areas. And one of the main things it does is it stores procedural memories. So if you play piano or you dance, you know that when you're first learning those movements, there's a lot of higher order thinking. You're thinking about where each finger should fall on the piano key. Or if you're dancing, where each movement of your arm, how that's supposed to turn while you're spinning and doing something like that. But the more you practice it, it gets integrated into the cerebrocerebellum, and you don't have to think about it anymore. Suddenly playing that piece of music on the piano, it's automatic. Okay, so that's the part, that's where the cerebellum is coming in to store that procedural memory. Okay, or the dance movement. You don't really think about it, you automatically do that. Okay, and the last portion there is the vestibulocerebellum. That maintains balance and controls eye movements, right? So controlling your eye movements, all of that's going to be important in balance as well. Okay. All right. Heading down the brainstem now. We're moving out of the cerebellum into the brainstem. This is a critical connecting link between the spinal cord and the higher brain regions. A lot of axons are moving through this area, have a pathway through this area from the spinal cord up to the cerebrum and down. It's considered the oldest region of the brain. So a lot of other animals, they all have this region of the brain, and how the mammals differed is that our cerebrum grew much larger over time. And we know it's the oldest region of the brain because it controls our life functions. So heart, blood vessel function, respiration, digestive functions. It keeps us alive. Okay, and it's continuous with the spinal cord. So a lot of that cross section and the arrangement of the gray and the white matter in the brainstem looks very similar to the spinal cord. Okay, also plays a role in regulating muscle reflexes, involved in equilibrium and posture. And then I'm going to get into the reticular formation within the brainstem. So we'll talk about that separately. Okay. Let's see. Let me make sure I got all the parts. I think I did. Okay. So along the brainstem, you have the pons and the medulla oblongata. Okay. So okay. I think what I have up here refers more to the medulla oblongata. It contains several nuclei of cranial nerves. So a lot of our cranial nerves branch off of both the medulla oblongata and the pons that you can see there. The medulla is important in heart function, okay, and actually modifying the heart rate. Um, the pons is what has these important respiratory control centers. Okay, the pneumotaxic area and the apneustic area, those two are in the pons, and the other one, the rhythmicity area, is in the medulla. Okay, so these are what are referred to as the primary respiratory control centers. So, for example, the medullary respiratory control centers that's where there's aggregation of these cell bodies that provide the output messages to your respiratory muscles. So it's within the medulla where that initiation, those action potentials come from to stimulate your respiratory muscles. And this sets your rhythm of breathing, right? But sometimes you want to slow down your breathing or you want to increase it, right? And so that's where the areas in the pons come into play. Okay. So, for example, you have inspiratory neurons. They fire to get you to inhale, right? And they stop firing when you exhale. Okay. But if you do a forceful expiration, then you have other expiratory neurons that fire. So to adjust that rhythm, again, the pons becomes active. So the pneumotaxic center, what it's going to do is it has axons that connect to the medulla, and it can switch off the inspiratory neurons. It actually inhibits them. So an inhibitory neurotransmitter comes and inhibits the inspiratory neurons in the medulla, okay, in that rhythmicity center. So therefore, it limits the duration of the inspiration, and if you do that, breathing inward, it slows down. So you just change the rhythm of your breathing. Now, the apneustic area in the pons, it prevents the inspiratory neurons from being switched off, right? And then that ends up providing an extra boost to the drive to inhale, right? So inhaling increases. Inspiration, inhaling, they're the same thing, okay? The pneumotaxic center actually dominates the apneustic center in order to halt inspiration and allow expiration to occur normally. All right, so make sure you kind of can come up with that sequence of events between your rhythmicity center, apneustic center, and pneumotaxic center. All righty. I think I have a note on here that it's on page 500 in your book, so that might be true. You might, if you can't find it, look on page 500. All righty. The reticular formation. So this is referred to as part of what stimulates the reticular activating system. Okay, so what the reticular formation is itself, it's a series of nerve cell bodies, the nuclei and their fibers, their axons that span throughout the medulla and the pons, and they actually span up through the midbrain to the thalamus and the hypothalamus. Okay, so it's this widespread network. And what it's doing is it's receiving the synaptic input coming up from the spinal cord. And it's integrating all of that synaptic input. And so that input, that sensory input, is going to come and stimulate the cortex. But just a split second before it does that, what happens is that network, again, by integrating the synaptic input, it sends a wave of depolarization ahead of the actual synaptic input coming in to promote alertness of the cortex. So what that means is, again, what we've studied, the alertness of the cortex, it means it's bringing those neuron cells closer to threshold. So that once that sensory information comes in, they're going to respond so much more quickly. So it just helps direct attention towards specific events, right? So what you're seeing there in the blue, the arrows, that's really showing what are called ascending fibers. So these are the axons that have action potentials coming from the spinal cord up to the cortex. Okay. So again, they're all interconnected. They kind of are widespread in all these different areas of the brain. Alrighty, so it just controls alertness, and it kind of wakes up the cerebral cortex that, hey, information's coming, get ready. So the faster you integrate and determine what that information is, the advantage you have in this world. Okay? That's it for the brain portion of the central nervous system. The next lecture will cover the spinal cord, and then we'll also get into how the spinal cord controls and integrates information from our internal environment, from our organs, things like that. We're going to get into the autonomic nervous system, and we'll go over the sympathetic system and the parasympathetic system. All right, thanks, guys.